Rapid Antigen Test for COVID-19 detection. COVID-19 is also called as SARS-CoV-2, which is an evolution of SARS-CoV-1. SARS means severe acute respiratory syndrome. And here we can see a rapid antigen test strip. In the extreme right hand side, we have a test spot. So where the sample is being spot over here, and there is a capillary action which I'll be explaining in upcoming slide. And we have a test line and a control line. Moving on to next slide. Types of COVID testing. So there are majorly three types of COVID testing. The first one is molecular or PCR, followed by antigen test and antibody test. The molecular or the PCR one is called as the gold standard to diagnose an active infection with COVID. And our major interest for today is antigen test, so which can diagnose an active infection with COVID. Again, so the sample is collected from the nasopharyngeal swab, as we can see over here, or it can be a throat swab. So it is always advisable to give sample to the expertise medical staff. Do not do at home because it leads to inaccuracy. Why it is called rapid test? Because we can get results within 15 minutes, and it is less expensive as compared to the molecular or the PCR one. Again, I'll be explaining in the further slide about that little bit. And it is less expensive as compared to antibody test itself. Antibody test is a blood test, so it is like post-COVID situation where the human body produces antibody against the infection, COVID infection. So it is the only way to diagnose a previous infection with COVID. It doesn't deal with the active infection as compared to antigen test and molecular PCR test. Going to next slide. Now the mechanism behind the antigen rapid test. So the mechanism behind this is lateral flow immunoassay. So why immunoassay? So we have an antigen. So our antigen of interest is nucleocapsid protein antigen, and we have an antibody. So fluorescently labeled antibody present in the conjugated pad. So it has a specific paratope which is specific against the nucleocapsid protein antigen. So this is the nucleocapsid protein antigen. And this is another antigen, but our major interest lies on the nucleocapsid protein antigen. Now, once the swab is collected, it is being dipped into the buffer or the neutralizing solution, after which a drop is put onto a test card. So this is the test card. Now, how it works? It works similar to a pregnancy test, where from the urine HCG is detected, human chorionic gonadotropin is detected. So here the swab, so the buffer solution having the swab is put as a drop over here. Now there is a capillary action, and because of which there is a lateral flow in this direction as it is shown in the arrow. Now once it moves from left to right, so there is a conjugation happening between the fluorescently labeled antibody which is specific towards the nucleocapsid protein, and it can mobilize. So it is a movable antibody. And further we have a test line and a control line which has immobilized antibody, which I'll be explaining in further slides. And absorption pad will restrict further mobilization of the antibody. Now it is very much important to interpret the results. So what and all the results we can get? We can get a positive result, we can get a negative result, we can get an invalid. The result is invalid. In a positive result, now this is the test card. The sample is put over here in this end, and first we have a test line and we have a control line. If we are getting a red band in both the cases, in test line as well as the control line, then the patient or an individual is detected to be positive, or he is infected with the COVID-19 infection. In case of negative, only in the control line we can see a red band, hence he or she is not infected with COVID. There can be many reasons for invalid results. like the sample which we have collected is not proper, the sample is degraded, or the quantity of swab collected, the process is not wrong. Sometimes it happens when we do it at home, and always it is preferred to use a single strip, not reusing it, which can lead to inaccuracy. So these are the three results to be expected and the analyzing way. Now moving on to next slide, where we'll go deep into the mechanism, like what is inside the conjugate pad, what is inside the test line, what is inside the control line. Here, once the swab is collected from the nasopharyngeal or from the throat, now this is inside the strip. What really it has? So it has antibodies. So this is the conjugate pad. The blue one is the test line, and here its orange one is the control line. So in the conjugate pad, as I had mentioned earlier, it has a fluorescently labeled antibody which can mobilize. In the test line, we have immobilized antibody, and in the control line also we have immobilized antibody, but it is called secondary antibody. Now, once the sample, if the sample having the virus, it is being put onto a test card. Now there is a capillary action over here. We'll move on to the second image. Now here we have an antigen, that is nucleocapsid protein antigen. Now there is a capillary action. It will move from left to right. There is a lateral flow. Hence, because of the specificity of the epitope and the paratope, now the antigen will bind to the fluorescently labeled antibody, and further it will move towards the test line and the control line. Now, as the antigen is binding to the primary immobilized antibody of the primary line, and further which is not binding to the test line, will move on to the control line. But control line is non-antigenic specific, so the fluorescent probe will bind to the antibody, hence it is secondary antibody. But in both the cases there is a binding of the in first case there is binding of the antigen, in the second case there is binding of the probe with the control line. Hence it is a positive test, because in both the cases we will receive a band. Now in case of a negative test, if for example in sample there is no antigen, a nucleocapsid protein antigen, then what happens? During the capillary action, the primary antibody which is immobilized in the test line is free. There is no binding happening here, because there is no antigen, no binding. But the antibody fluorescently labeled will move because of the capillary action, and it binds to the control line. Hence there is only one red band being seen over here, and others which are not bound is absorbed by the, here we have absorbent pad. So because of only one red line, it is a negative test. So this is the basic mechanism. Now we'll move further on what are the merits and the demerits. Yes. In the merits of the antigen testing of COVID-19, it can diagnose an active infection, as I had mentioned in the second slide. And again, the results which we are getting is like within 15 minutes, and again it is less expensive. These are the major merits as compared to molecular PCR or the antibody. Now, what are the again, there are different companies like PanBio, or different companies, they produce strips now. Nowadays it is available as commercial. Like from home also we can order it and we can test it. Again, it is advisable to do a test wherever there is a expertise medical staff is there. Now I'll be moving on to what are the demerits. The demerits of COVID-19 swab test or the antigen testing of COVID-19 is, this test is highly specific but not as sensitive. This means a positive result is almost a true. Whatever, that is a patient who is having a COVID and he has diagnosed positive, hence it is highly specific. But sometimes they get a negative result which is false. So what is sensitivity and what is specificity? Sensitivity deals with only infection, like the affected individuals, and specificity will deal with the healthy individuals. Now, in sensitivity, it is the ratio of number of true positives and the number of those who are infected. That is true positive plus false negative. Here, true positive plus false negative. In case of specificity, which is the ratio of true negative and true negative plus false positive, that is the total people who are not infected. Now, it is being told, like it is also being seen like it is highly specific in the sense the number of true negative is high on the people. So the people who are healthy, for sure they'll get a negative result. There is no deviation in that. But in case of sensitivity, what happens is there is a decrease, there is a high decrease up to 50% decrease. Like specificity read in one paper was 95 to 97.6, but sensitivity is 55 to 57. Again, it will depend on various strips used, but it has been seen that sensitivity is very, very less. Because as sensitivity is less, so here we can see the people with COVID-19. So as sensitivity is less, the false negative is it's very high. The number of false negative, that is the people actually they are infected, but even though they are infected, it is showing a negative result. Hence, it is called a false negative. In order to avoid this, hence molecular PCR is a gold standard to diagnose active infection with COVID, but it takes more than 24 hours as molecular PCR depends on the mRNA, the genetic material, but the antigen test, it depends on the nucleocapsid protein antigen, that is the surface, the protein which surrounds the COVID-19. Hence, it is the best and easy way is the antigen test of COVID-19. But the major demerit is because of the major demerits of sensitivity, molecular PCR is widely used for a higher accuracy.